guys, thanks for joining me in the closet here at the library. Wanted to talk a little bit about Mendel. Hopefully you've done a little reading on that, thought about it. Maybe you've even looked at the PowerPoints that I have linked to your Redline site. But let's do a little practical look at it. Okay, so the first thing that Mendel discovered when he was busy crossing peas in the, oh gosh, lost that. He was in the labs, no, he was in the greenhouses, yeah, that's it. He was in the greenhouses in the monastery playing with peas, trying to see what would happen if he cross-pollinated these peas, and he came up with a few things. The first one was the concept of recessive and dominant alleles. So, if we took purple and we crossed them with white, he found that the F1, that first filial generation, was all purple. And if he crossed two of the offspring of this first generation and made an F2 generation, he came up with a 3-to-1 ratio of purple to white. And this is where he got this concept of dominant and recessive. And so if we were to use the letter, I really don't like the letter P, so I'm going to use the letter R instead, just to mess with you. So we're going to use capital R to represent dominant and little r to represent recessive. In the first generation, he crossed something like this. We must use the same letter, otherwise we don't. The concept of dominant recessive is shot. We don't want to use two different letters at this point. And when we cross those, we use our simple Punnett square with the 4 boxes, and we have to segregate the alleles. And you should recall from meiosis that you can only give one allele or the other. You cannot give both. Otherwise, you have quite a messed up offspring. And so when we do this cross, we end up with all offspring that have a heterozygous genotype, big R, little r. And their phenotype is all purple, what they look like. Now when we take two of these individuals and cross them, and I'm going to sneak this in right here, okay, our Punnett square comes out like this. We segregate, and we end up with some very different individuals. Okay, and there we go. So we have one homozygous dominant, they're both dominant alleles, purple. 2 heterozygous dominant, so they have different alleles, they're also purple. And 1 recessive homozygous individual who is white. Hey guys, board's all erased, ready to go. I'd like to talk to you about what do you do if you have more than one trait that you're studying. So this is where Mendel came up with the idea of independent assortment, that alleles segregate independently of each other. Let's look at some peas, shall we? So if I'm going to cross a green round pea that is heterozygous for the trait with a yellow wrinkled pea. Let's write that out. So I said it's heterozygous, green's dominant, so I have to have at least one big G, and I said it was heterozygous, so it must be big G, little g. And round, I'm going to use the letter R, big R, and I said this was also heterozygous, so it has to be big R, little r. And I'm going to cross it with that yellow, and I'm going to use little g for yellow, because I have to use the same letter for color, and I'm going to use little r for wrinkled, little r, little r. Both plants must have 2 alleles for the trait. And now we know that they can only give one of each of those alleles to their offspring. Now, my Punnett square is going to have to be a little bigger though, and so for these types we're going to need 16 squares. So we're going to make our giant Punnett square. And there we go. Now to segregate independently, what has to happen is this big G could be donated with this big R. I'm going to put that right there. Also, this big G could go with the little r. Okay, that's 2 gametes down. Now, the little g could go with big R, or little g could go with little r. Look at that. I have 4 different possibilities of gametes that could be made from this parent. This other parent over here only can do little g, little r. Please note that each gamete here has to have at least 1 G and 1 R so that the final offspring ends up with 2 for color and 2 for shape. And then we bring down and over and we end up with big G, big R, little r. Okay? And then over here we have big G, little r, little r. Totally different. Then we have little g, big R, little r, and little g, little r. I hope that you can see that each row would be exactly like the first row because there is no variation in the parents' gametes on this side. So we have different phenotypes and genotypes. This individual is heterozygous for both traits, just like the parent, would be green and round. This offspring is heterozygous for green and homozygous recessive, so wrinkled, green wrinkled. Then we have a yellow, little g, with a round shape. And then finally we have a yellow wrinkled, like the parent. So we have different phenotypes that could only be produced if the gametes, the alleles were segregated independently of one another. Now I need to erase the board again so we can move on from here. Alright guys, so here we go. Now we're going to look at a trihybrid chorus. That's right, 3 traits we're going to look at. So in this case, this is a similar cross from your book. We have purple flowers, yellow seeds that are round. Okay, and we're crossing that with purple flowers. Ooh, what's the antithesis of yellow? Don't know. Not yellow and wrinkled. So let's see what happens. Do we really want to do a Punnett square for this one? I don't think so. That's about 64 boxes. I get tired just thinking about it. Let's just look at probability. What is the probability that we will get an offspring that has this genotype? Little p, little y, big R, big R. So what we have to do is determine for each parent what's the chance that they're going to give that allele. Okay, so if we look at this first parent, what is the chance that this parent who has a big P and a little p is going to give a little p to this offspring? That would be 1/2. Do you all agree with me, I hope? And then what's the chance that the other parent will also give little p? That's also 1/2. So I multiply 1/2 times 1/2, but we're not done. Now we have to look at the seed. What's the chance that this parent is going to give a little y? So we have big Y or little y, so it is, again, 1/2. We look at the other parent, what's the chance they're going to give a little y? Well, it's 100%, so we're going to multiply that by 1. And then finally we look at this round shape. What's the chance that this parent is going to give a big R? Well, that's a 1/2, right? And then we come over here, and what's the chance that this parent's going to give little r? That would be 1. Then we multiply that all out, and we get, let's see, 2 times 2 is 4, times 2 is 8, times 2 is 16, 1/16. That's how you solve your probability crosses. Oh, hi. I was just realizing I was looking at chapter 15. We get into what Sir Thomas Hunt Morgan figured out about linked genes and chromosomal inheritance. And I just wanted to go over sex-linked inheritance just a little bit. So this is the rest of the information we've done so far is chapter 14. This would be chapter 15. And Morgan, he worked with fruit flies. And he did a lot of experiments breeding them, and he discovered that certain traits always went together. They're linked. But specifically I want to look at sex-linked. What does that mean? A sex-linked gene is a gene that is on the X chromosome. And so all you guys out there are probably saying, why is it on the X? Why not on the Y? What's wrong with the Y? Well, girls don't have a Y. So if it's important for both members of a species to have that gene in order to survive, it really needs to be on the X chromosome. Let's go back and remember, how do we write the genotype for a girl would be XX, okay, and the genotype for a boy is XY. So if we have something, a trait that's only on the Y chromosome that is important for both members, we got a problem here because the girls don't have a Y. So we will see that there are certain genes that are carried on the X chromosome. One of those would be color blindness. And so we're going to write color blindness with a B for blindness rather than a C, because it's easier to see if it's dominant or recessive. And if we have a girl who is heterozygous, okay, so she has a big B and a little b. Please note I've superscripted those. So they're carried on the X. That's how I denote that. And I'm going to have her have children with a guy who is not colorblind. Big B. He's not colorblind. That's it. And they're going to have offspring. Let's go back to that good old Punnett square and see what happens here. So we have to segregate the alleles just like we did. So we have the female here with X big B and X little b segregated, and the dad segregates his sex alleles. And now we have to bring down and over and figure out if we have boys or girls and if they're colorblind or not. And we have the following individuals. So let's look at each of them. This girl right here, she's XX, so that means she's a girl, and she has two big B's, if you will. They're both dominant, so she is not colorblind at all. And we have another female over here, but she's heterozygous like her mother for color blindness. Over here we have a boy who is not colorblind. He's got a big B. And finally we have a boy who is indeed colorblind. He has a little b. And so you notice that out of this, only one of the children was colorblind, and it was indeed a boy, because he has no way to hide it, if you will. Because remember now, the dominant gene will basically hide a recessive gene, but because he only gets one allele, it has to be expressed. There's no choice. Girl up here, she can hide it. She has a big B, so to hide the color blindness. But notice that mom, who was the same, did indeed give it to her son. So she is called a carrier. Mom is a carrier of a recessive trait. She doesn't have it, but she passes it on to her offspring. And this is why males tend to have all of any disorders that are sex-linked. They are more likely to have, because they only have one shot at it. And so there we go. There's sex-linked traits. See you later.